Good morning to all of you. Today, in your genetics syllabus, I am going to discuss with you all about inversion. In the previous video, I have discussed about your deletions and duplications. So, let us see about the inversion. After this lecture, I will discuss about the translocation and the human karyotype analysis. So get ready for the next videos also and study them well. So about the inversion simply involves rotation of a part of a chromosome. It simply involves rotation of a part of a chromosome or set of genes by 180 degree. So 180 degree it gets rotate. A part of a chromosome will be broken down. Obviously, two breaks will occur and that broken fragment will be rotated 180 degree on its own axis and it will again rejoin. So it essentially involves occurrence of breakage and reunion within the same chromosome, and the net result of inversion is neither gain of a chromosome fragment, neither loss of a chromosome fragment, or you can say neither gain or loss of a genetic material. So here nothing will be gained, nothing will be profit or loss will occur, only the sequence of the chromosome will be little bit changed. So here you can see this chromosome having this configuration A, B, C, D, E, F, G, H, I, J. Now, in this chromosome segment within the F and between the F and G and between I and J, there creates a break. So, two breaks occur from this side and this side, and that fragment which contains G, H, I, it rotates to 180 degree and looks like this. It becomes now IHJ. That part is your inverted segment. So in this way your inversion occurs. Now how it is possible you will see later them on this video. Now inversion are of two types, can be inversion, paracentric inversion, pericentric inversion, and obviously you have a inversion heterozygote in which only one chromosome will be only one chromosome from the homologous pair of chromosome will be involved in the um, formation of your inverted segment, while the other chromosome is in the normal order. So it is saying here an inversion heterozygote has one chromosome in the inverted order while its homolog chromosome will be in the normal order. So those who are inversion heterozygote, they have we have homologous pair of chromosome for each chromosomes. Now in inversion heterozygote, one of the chromosome will have the inverted segment while the other chromosome is normal. That's what inversion heterozygote means. And the location of the inverted segment can be detected cytologically also in the meiotic nuclei of such heterozygote by the presence of an inversion loop in the paired homologs. So, to observe inversion in case of inversion heterozygote in which one of the chromosome have the inverted segment while its own homolog, that means the other chromosome from the homologous pair of chromosome, they are normal. It can be observed cytologically, it can be observed through the help of microscope, especially in the meiotic nuclei during the meiotic cell division by observing a loop-like structure in within the paired homologs. So see So this is a inversion heterozygote. You can see them clearly that one of the chromosome is normal from that homologous pair, while the other chromosome having that inverted segment, it has this inverted segment present. So this is called inversion heterozygote. And how they arrange themselves during meiosis, during the packaging stage of meiosis, it will be look like this. This is a simplified diagram. I'm trying to zoom in. So you can see here that it forms a inversion loop. A inversion loop is paired. Inversion loop is created so that it can pair with its homologous chromosome, right, within the inversion heterozygote. So you can see this upper chromosome is normal, A, B, C, D, E, whereas the chromosome having the inverted segment, they tries to pair with its homologous chromosome, as it is its chromosome segment is in the inverted order. So that's why they form a loop like structure like this, and thus they are pairing. Like you can see, they are trying to align themselves with its homologous chromosome, with its homologue within the homologous chromosome, and that's why the loop like structure has been observed. So in case of inversion heterozygote, it is possible to detect cytologically, because you can see a characteristic loop like structure is observed trying to pair with its homologues. So I think you have able to understood this part. I am going more further. Now, two types of inversion can be observed. One is called paracentric inversion, another one is called pericentric inversion. Most of the case, the difference between paracentric and pericentric inversion comes in your exams. Um, Let us see what it is, what it does it mean. So the location of the centromere relative to the inverted segment will determine the genetic behavior of the chromosomes. So depending on whether the inverted segment contain the centromere or whether the inverted segment is out from the centromere, depending on that, inversion are of two types, and it is saying that what will be the genetic behavior of the chromosome that fully depend on your inverted segment having the centromere or not. Now if it is saying clearly that if centromere is not included in the inverted segment, that is called as paracentric inversion. So paracentric inversion is such inversion which occurs on the arms of the chromosome. The inverted segment are found within the arms of the chromosome, but it does not include your centromeric region. Whereas in pericentric inversion, the inverted segment includes your centromere. That is known as pericentric inversion. So when inversion includes your centromere, that is called as pericentric inversion. Just look at the diagram for you to clarify your doubt. So in paracentric inversion, you can see the inverted segment. One inverted segment occur. See the above figure, okay? One inverted segment occur in this region. One in another inverted segment occur in this region, right? But this inverted segment does not includes your this centromere, which joins the two arms of the chromosome. Centromere is a fully heterochromatic region. Actually, it is a fully inactivated region. Here you will find out the DNA, but that DNA will not code for any functional protein. It is fully heterochromatic regions, fully coiled regions, which joins the arms of the chromosomes. Now if the centromere is not involved during the inversion process, it simply means that the inverted segment does not contain your centromere. That is known as paracentric inversion. But in the second case, if you see the next diagram, this one, the second diagram, the inverted segment includes your this centromeric region. That's why it is called as pericentric inversion. So you can see the arrangement of the chromosome in the inverted order, but the it also includes your centromeric region. That's why it is called as pericentric inversion. And in both cases, when when the cells are going to be divided, when the chromosomes are going to be distributed to the daughter cells, a characteristic cytological observation will be detected during your paracentric inversion as well as in your pericentric inversion. Here also you will see some sort of bridge breakage fusion cycle that we have seen in case of duplications as well. Here you also we will see these things. So this was about your paracentric and pericentric inversion. Let us, I think you have understood this part. Now next part that I will going to I want to do to you all is this one. So it is saying here that your homologous chromosomes with identical inversion in each member pair undergoes normal distribution in meiosis. Now, if your homologous chromosome, that means the pair of chromosomes, one comes from mother, comes from father, if this homologous chromosome have same type of inversion, if these homologous have chromosomes have same type of inversion, then they will easily distribute, normally they will distribute during meiosis process, because at identical location both the chromosomes are having the inverted segment, so there will be no any problem. Like for example, uh, the like for example, suppose this is one, okay, wait for a minute, this is one chromosome from the homologous pair having the sequences A, B, D, C, E, F, G, H. Okay, and this is another homologous. This is another chromosome. This is another homolog pair of the chromosome having the same sequence, same inverted region A, B, D, C, E, F, G, H. So they are having the same inverted segment, right? That is a DC. So during meiosis, when these chromosomes, these chromosomes from the homologous pair, they will undergo normal distribution. They will be easily migrate to the opposite poles distributed among the daughter cells. Okay, so it cannot able to detect cytologically in case of chromosomes having both the sets are having identical inverted segment. But problem occurs when inversion heterozygotes. Problem occurs in case of inversion heterozygotes. What happen in inversion heterozygotes? I have already said about inversion heterozygotes, that in inversion heterozygotes one of the chromosome will have the inverted segment and its homologue will have a normal chromosome. That means in inversion heterozygotes, this is the inversion heterozygotes that I want to show you in the diagram, that one of the chromosome will have the inverted segment, that is A, B, D, C, E, F, G, while the other chromosome is normal. Other chromosome from that homologous pair is normal, A, B, C, D, E, F, G. So this is called inversion heterozygotes, and inversion heterozygotes, when it undergoes during meiosis process, it will produce some deletions, some duplications, and other curious configurations will also be observed. Okay, So this is the problem that we face when we will see them cytologically when we are when chromosomes are having inversion heterozygotes, right? So it is saying clearly that homologous chromosome having identical inversion in each member pair undergoes normal distribution. Okay, but when crossing over occurs in inversion heterozygote, that means one of the chromosome from the homologous pair is having inverted segment, the other chromosome from that same homologous pair will is normal. So in case of inversion heterozygote, when crossing over occurs, it produces deletions, it will produce duplications and other curious configurations. You will see them just a minute after, some few minutes after, about the image, that what happens in inversion heterozygotes. Now all these inversions can be readily be detected by comparing of linkage maps of normal and inverted chromosomes. So inversions, all the inversions can be easily be detected by comparing the linkage maps of the normal and inverted chromosome. Actually, linkage map shows the relation of the genes, relation of the alleles that are located on the chromosome, how closely they are linked, how closely they are placed, and more linkage between the genes, that means they will be segregated as a unit during your chromosomal division. So it is saying that when genes are found on different chromosomes or far apart on the same chromosome, they assort independently due to crossing over and are said to be unlinked. As we know, Mendel's law of independent assortment that you have learned in your class 12, that has been mentioned that genes or alleles tend to segregate independently. But if the genes are close enough on the chromosomal arm, they can inherit it together as a unit, right? They are said to be linked. But when genes are found on different chromosomes, if genes are found on different chromosomes, or if the genes are placed far apart on the same chromosome, these genes will assort independently, they will segregate independently due to crossing over and are said to be unlinked. But if genes are very close together within the same chromosome, say the genes are located very close, okay, here and here within the same chromosome arm, then what happens if the genes are very close together on the same chromosomal arm? They are said to be linked. They will move together during your cell division as a unit. So it means that alleles or the gene versions already together on one chromosome will be inherited as a unit more frequently. So if genes are placed very closely within the chromosome in the same chromosome arm, then what happened during cell division? They will be together. They will be present together on one chromosome, and they will be inherited as a unit. That type of genes are said to be linked, whereas unlinked genes are placed far away from the chromosome. There is a more chance of for them to assort independently. They will segregate independently due to crossing over. Okay, so it simply means like this. I just remember the crossing over that occurs between the chromosomes, the crossing over that occurs between the chromosomes. So So if this is the two chromosomes, and I want to say that if genes are located close together on a chromosome. So if I say these two genes, I not name them, so there is a chance, there is no chance of crossing over between them, there is no chance of separation of these genes, because they are at the same location, they are very, they are close, very they are linked very closely, so there is no chance. But if suppose if the genes are the genes are placed here and here, so you can see there is a chance of crossing over at this region, so these two genes will segregate independently. That's why the phenomenon of linkage and non-linkage occurs. So through the help of linkage maps, that means to see that if the two genes are close enough, that means if the two genes are linked or not, and how tightly they are linked. Okay, we can observe them by using the data from the genetic crosses. We do crossing experiments, and we get the data, the information related with the genetic cross, and we calculate a term called recombination frequency. We calculate a term called recombination frequency, or RF. So, to see that whether the genes are are close enough or not, we can, and how tightly they are linked, so that they can be segregated together as a unit during crossing over. For that, we have to calculate your recombination frequency. So by finding out the recombination frequency, it is simply recombination frequency is simply the statistical data of the different type of crosses, genetic crosses, that are used to see that how closely the genes are linked. So by finding out the recombination frequency for many gene pairs, we can make linkage maps, and we show the order and pattern of the genes and how the genes are placed within the chromosome, right? So that's, in this way we can see that we can detect the inversions as well, that by finding, by studying the linkage maps, because linkage maps find out the distance between the gene. It shows the order of the genes and relative distance of the genes on the chromosome. And thus in this way we can also, with the help of linkage map, we can find out the inversion as well, because in inversions the arrangement of genes will be in the reverse order. We can able to calculate and see them. So that's why what all written about in this part that I have said, just go through this part. And next is your cytological observation of paracentric and pericentric inversion. So it is saying that when uh, during pericentric inversion crossing over occurs, what are the things, what are the events that are going to be observed? Let us see. So, crossing over in heterozygous pericentric inversion, that means including the centromere, the inverted segment having the centromere. So, crossing over in heterozygous pericentric inversion results in deletions and duplications, and also produce rod shaped acrocentric chromosomes. So just like I have said that in case of heterozygous pericentric inversion, that means one of the one of the chromosome from the homologous pair is having inverted segment, and it is pericentric, that means it includes the centromere, right? It includes the centromere, while the other chromosome from that same homologous pair is normal. So when crossing over occurs in heterozygous pericentric inversion, it will cause you deletions, it will cause you duplications, and also produce acrocentric chromosomes, that means the chromosome fragment without any centromere. The first meiotic the first meiotic anaphase figures appears normal. Now, w- during crossing over, when they go through the first meiotic anaphase, as we know that meiosis occurred two times, meiosis 1 and meiosis 2. So when they undergo, after crossing over, when they undergo first meiotic anaphase, it appears normal. But the two chromatids of the each chromosome usually have arms of unequal length. During the first meiotic anaphase, when crossing over, so first crossing over in heterozygous pericentric inversion occurs, and after that you will see that during the first meiotic anaphase, when the chromosomes are separating to the opposite poles, you will see the two chromatids of each chromosome, they will have arms of unequal length. They will have arms of unequal length depending upon where the crossing over has occurred. So during your packaging stage of meiosis, you see the two chromosomes are involved in crossing over. The chromosomes are involved in crossing over, and if they are having heterozygous pericentric inversion, then you will see that during meiotic anaphase, the two chromatids of each chromosome, they will have arms of unequal length when they are getting separated to the opposite poles during anaphase 1 of meiosis, and depending upon where the crossing over had occurred, you will get that unequal arm. Half of the meiotic products, that means the gametes, the polygams that are having the chromosomes, so half of the meiotic products will be non-functional and inviable, that means inactive, due to the presence of duplications or deletions in them. So it has been saying that when crossing over occur in heterozygous pericentric inversion, there will be deletions and duplications during crossing over, and thus the chromosome will have arms of unequal length when you observe them in the first meiotic anaphase of your cell cycle. And half of the meiotic products, after your meiosis process is completed, they are distributed in the gametes, because we know that meiosis occur for the formation of the gamete cells. So half of the meiotic products, that means the gametes or the polygons, whatever we are saying, they will be non-functional. So half of the meiotic products will be non-functional because the chromosomes within those gametes, within those polygons, having deleted segment, having duplicated segment within them, and the other half will be functional. Okay, other half will be functional. One quarter, that means out of four, one will have a normal alignment of chromosomes within the gamete, while the other one quarter will have a inverted segment. So this is a condition that will be observed when crossing over in pericentric inversion takes place. Now, theoretically, I have said, let us try to explain them in diagram. So this is a normal chromosome that you can see. The upper one is a normal chromosome, while the lower one having a inverted segment, the ABCD, ACBD. So it is having the pericentric inversion. That means the inversion having the centromere involved. So this centromere is involved, which is joining the two arms of a chromosome. So you can see the pericentric inversion here, that is the inversion having centromere, inverted segment having centromere. And when this heterozygous pericentric inversion is involved in crossing over during meiosis of your cell cycle, this is what will happen. What will happen? Let us see. So obviously I have said that during heterozygous inversion, when inversion occur in heterozygotes, that means one of the chromosome is having the inverted segment while the other chromosome is normal, there will be formation of a loop like structure. So a loop like structure has been observed here, tries to align with its homolog pairs, try to align with its homologous segments within the same chromosome. And you see them, the two chromosomes are aligning themselves. That's why a characteristic loop like structure is observed in order to align themselves properly during synapsis. And the point of crossing over shows here in this region, where you can see that now this point of crossing over can occur in any places, depending on that it will result in deletions, it will result in duplications and rod shaped chromosomes which are acrocentric chromosome. Some fragments will be observed which will not have any centromere. Okay, so depending upon where the crossing over has occurred, it may occur at this position also, it may occur at this position also. Depending on where the crossing over had occurred, um, it will lets you to form chromosome arm having unequal in length. So here we can see in the left hand side it shows that end of meiosis 1, what will be the products, chromatid products that it will produce while during crossing over between in the heterozygous pericentric inversion. So this is what you will get. The first one is the normal segment, while the second one will having an inverted segment A B C A. Okay, and you can see clearly that there is a missing of D segment. So the D is missing. D is missing. D is missing in this second chromosome. So there is a deletion of D, but there is a duplication of A segment. So you will get chromosomes of unequal length. They will have an arm of unequal length. They will have deletions and duplications during your first meiosis, when the first meiosis process is over. See the second chromosome, you can see D, B, C, D. That means the A segment has been lost while the D segment has been duplicated here. And the last chromosome, you see the whole chromosome is being in inverted order, A, B, C, D. Okay, A, C, B, D. Now, when these chromosome pairs are involved in second meiotic cycle, that means meiosis 2, this is what it will occur. You can see the upper chromosome is normal, so it will be distributed to the gamete, and that gamete will be a normal product. While the second chromatid, when they are getting distributed, this gamete, the gamete which will receive this chromosome, the gamete which will receive this chromosome, they have a duplicated A segment while a deleted D segment. So duplication of A segment and deletion of D segment, that can leads to some non-viable gametes. And you can see the third one when it is distributed in the gametes during the end of meiosis 2, there is a duplicated D segment but deleted A segment. And the last one will have a whole inversion product that we can see. So you can see here clearly that that half of the meiotic products are non-functional, half of the products are meiotic products are non-functional, that is 2 out of 4 are non-functional. If you cross them you will get 1 by 2, that means half of the meiotic products are non-functional because there is a presence of duplications, there is a presence of deletions within them. So when they are getting distributed in gametes, the gametes become non-viable. While the other half, that means this one and this one, the other half will be functional, but one of them is normal. That means out of 4, one is normal. That is this one. While the other quarter, that is 1 by 4, will have a chromosome with inverted segment. Right? So this was about the arrangement of the chromosomes when they are in the pericentric inversion. Now we will discuss the crossing over in case of paracentric inversion, and that's a very interesting thing. I hope you have understood this much. Okay, just comment me in the WhatsApp group if you have any doubt. So let us see about the meiotic products that will occurs in your during crossing over in paracentric inversion. And during paracentric inversion, the inverted segment does not involve your centromeric region. And here we are discussing, and here we will see the crossing over during your heterozygous paracentric inversion. That means one of the chromosome will have the inverted segment but does not involve the centromere, while the other chromosome of that same homologous pair is normal. Okay, in previous one that it was the inverted segment including the centromere, but here you will see what happens when we are doing crossing over in case of heterozygous paracentric inversion. That means one of the chromosome will have the inverted segment but it does not involve the centromere, while the other chromosome from that same homologous pair is normal. So a crossing over in inverted region of heterozygous paracentric inversion will produce a dicentric chromosome. A dicentric chromosome is a chromosome which will have two centromeres, which will forms a bridge from one pole and the other to the other pole during the first anaphase of meiosis 1. So, when crossing over occurs within the inverted segment of a heterozygous paracentric inversion, it will produce such chromosome having two centromeres known as dicentric chromosome, and it forms looks like a bridge. It forms a bridge from one pole to the other pole during the first anaphase of your meiosis 1. I think you have understood what I am trying to say. I'm repeatedly saying heterozygous paracentric inversion means one of the chromosome from the homologous pair having the inverted segment but does not involve the centromere, while the other chromosome from the same homologous pair is normal. So when crossing over occurs in case of heterozygous paracentric inversion within the inverted segment, then what happen? It will produce a dicentric chromosome. A dicentric chromosome is a chromosome having two centromeres, and it will looks like a bridge. A bridge will formed from one pole of the chromosome arm to the other pole during the first anaphase of meiosis 1. Obviously, this bridge will not remain for a long time because the spindle fibers will try to migrate the two chromosomes to the opposite poles. So, the bridge will eventually rupture, it will break somewhere along its length, and it will result in such fragments that will contain duplication or deletion, or both duplication and deletion will be found, as we have seen in paracentric pericentric inversion, that when crossing over occurs in heterozygous pericentric inversion, the chromosomes are having duplicated segments, having deleted segments as well. Here also you will see the duplicated segments or deleted segments, but uh, important cytological observation that we will see a bridge. That bridge is forming as a result of dicentric chromosome formation, chromosome having two centrosomes, sorry, so not centrosome, centromeres, right? In case of an acentric fragment, suppose somehow an acentric fragment has been formed as a result of crossing over in paracentric inversion heterozygotes, as a result of crossing over during pericentric inversion also. If acentric fragment occurs, that means chromosome having no centromere, that fragment unluckily will be lost from the chromosome because it will fail to move to either pole because they don't have any centromere. How it will attach with the spindle fiber, how it will migrate to the opposite pole. So these acentric fragment, as they don't have the telomeric regions as well, it will be unstabilized, it will be break down somewhere and will not be included in any meiotic products. That means it will not found in the gametes. Heterozygous for paracentric inversion are highly sterile and produce only parent-like progeny, as half of the meiotic products are non-functional. Now it is very serious that if crossing over occurs in heterozygous paracentric inversion, we will form a bridge-like structure, and that bridge will eventually will break, and it leads to such products, such chromosomal products which are distributed among the gametes, that half of the gametes are non-functional while the other half will be functional, that will bear only parent like progeny, it will have the parent like features. So let us see that how we can detect your crossing over during paracentric heterozygous paracentric inversions. So let us see. So you can see I have shown just one arm of your each chromosome from the homologous pair of chromosomes. Well, you see that one of the chromosomes is having the inverted segment ADCB, while the other chromosome is normal. That's why we call them as heterozygous paracentric inversion. So crossing over will be occurring between them during your packaging stage of meiosis 1 of cell cycle. So here it is saying pairing of between homologous chromosome. So that's why inversion loop will also obviously it will be observed. So you can see a characteristic inversion loop that tries to pair what with its complementary with its homologous chromosome. Okay, and crossing over is occurring here. Okay, it can occur in any place, okay, no problem, but it is occurring here. So what will happen after crossing over? Exchange of the chromosome segments, obviously. And during exchange of chromosome segments, you will see that during your anaphase, during metaphase, during anaphase especially, you will observe a dicentric chromosome, a dicentric bridge will be observed. uh, a dicentric bridge will observe. You can see here that this arm is joined by centromere, and this is another centromere joined by this arm and this arm. And you also get an acentric fragment. This fragment will be eventually be lost from the chromosome because this fragment does not have any centromeric region. This fragment does not have any centromeric region. This one, right? And how this dicentric bridge is formed, if I show you, you can able to imagine. Okay, let's get started now. So you can see your this one a first is centromeric region, then A, B, C, D, E. Now this part, again I repeating, centromeric region A, B, just follow the line, C, D, E. Okay, so this line represents this one, this one. Okay, now the centromere is joining the two arms. Let us see the second one. Again centromere A, B, then crossing over has taken place. C, D, A, centromere. A, B, C, D, A. So it will be looking like this. A, B, crossing over, C, D, A. That's why it looks, this is the second one. You see, A, B, C, D, A. Then again, centromeric region. Look at the third one. Look at the third one. A, D, C, B, E. A, D, C, B, E. A, D, C, B, E. This is the third one. A, D, centromere, A, D, C, B, E. So that's why in this way we get a dicentric chromosome. Dicentric bridge occurs. and it will break randomly also somewhere. And the last one is your an acentric fragment that we will get. Okay, a acentric fragment we will get. uh, this is the acentric fragment. If I hope, okay, this one, I am zooming it. Look at this line. E, not this line, sorry, not this line, it will be this line, the fourth line. E, B, crossing over, C, D, E. I'm repeating again. Let's look at the fourth line. E, B, crossing over take place. C, D, E. So this is the acentric fragment that occurred. That is a fragment, chromosome fragment without having any centromere. Okay, this is that this fragment. So this acentric fragment will be eventually lost. They will fail to pair to the chromosomes and will fail to pair to distribute among the gametes. So as a result of which random breaks will occur. Here I have shown the breaks in these regions between D and A, and that's why we get this one. We get this one where there is eventually a duplication, a deletion product we will get. And when your meiosis 2 occurs, when meiosis 2 will occurs, during meiosis 2 of your cell cycle, when the all the chromosome products will be distributed among the gametes, you see we will get one normal product, one deleted product. Means the E is missing. Okay, the E segment is missing in this product, where you can see a deleted product and inversion product we will get. So eventually you will see that in this uh, when we are doing crossing over during heterozygous paracentric inversion, in this case you will see a acentric fragment that is eventually lost. And heterozygous for this paracentric inversion are usually sterile, and it will produce only parent like progeny. So you will get only parent like progeny here, you can see here. And half of the meiotic products are non functional. Okay, so this was all about the paracentric and pericentric inversion crossing over. This type of questions may come if normal questions that is given in your exams, if come in your genetics examination. But nowadays normally we go for MCQ patterns due to this pandemic period. So they may give you an image, they may give you an image and answer the questions MCQ through MCQ. Okay, so after this unit is completed, I will try to give you some of the MCQs, some short questions. You have to answer them by studying those videos. Okay. Now let us see inversions in natural populations. So now we have to see practically that what will happen when inversion in natural population occurs. It is saying that in natural populations, pericentric inversions are less frequent than paracentric inversions. So these pericentric inversions, they lessly occur. Okay, that is inverted segment having centromere. So pericentric inversion does not occur frequently as compared to the paracentric inversions. Paracentric inversions appears to be very frequent in the natural populations of Drosophila, which is your fruit fly. For example, more than 31 different inversions are known in chromosome number 3. So most oftenly we find paracentric inversions, that is inverted segment without having centromere. This type of inversions are mostly found, especially we have seen in Drosophila, the fruit fly, Drosophila melanogaster. They have 31 different types of paracentric inversions within their chromosome number 3 only. Detection and detailed analysis of inversions are relatively very easy in Drosophila due to polytene chromosomes. Now, why we are studying so much in a small insect of fruit fly called Drosophila melanogaster? why we are studying the inversions there, because it is very easy to study the chromosomes of Drosophila because they have polytene chromosomes. What does polytene chromosomes mean? This will also be explained in separate video, but just I am trying to say that about the polytene chromosomes in little bit briefly, that polytene chromosomes are formed when multiple rounds of replication occurs and produce many sister chromatids which remain fused together. Like if this is a centromere, there is one chromatid, there is another chromatid, there is another chromatid. So there are many sister chromatids fused together to form a polytene chromosomes, which can be easily visualized in your microscope. And it occurs when multiple rounds of replication takes place, okay, that produce many sister chromatids fused together. That's why we can study the Drosophila parting chromosomes, and we can also able to study the detailed analysis of inversions within the Drosophila. About 31 different type of paracentric inversions occur in chromosome number 3 of Drosophila. And these polytene chromosomes are mainly found in the salivary glands of Drosophila melanogaster. So for observing the polytene chromosomes, we have to use the salivary gland. We have to sacrifice Drosophila melanogaster, and we have to prepare the sample mm, by taking their salivary glands. We have to use certain stains, certain dyes, okay, in order to visualize their chromosomes. And for visualizing the inverted segment, we have to go for karyotype analysis. We have to go for the banding techniques. Now, in other organisms, instead of in place of Drosophila melanogaster, in other organisms there may be frequent, but their detection is generally not possible without packaging analysis of appropriate crosses. So it means to say that in other organisms, if you want to study their inverted segments, if you want to analyze and detail study their inverted segments, then we have to see their packaging analysis. We have to see their packaging stage, okay, and that is not possible in all the cells because the cells are actually continuously they are doing their cell cycle. For this reason, we have to use certain chemical in order to freeze that stage, and that's why we use the spindle fiber disruption by using colchicine. Okay, different type of chemicals are being used in order to make the cells freezed in that state by using different chemicals. Like colchicine disrupt the spindle fibers, so the chromosomes will not able to migrate to the opposite poles. It will remain in the metaphase stage, and in this way some chemicals are used to detect the packaging stage of the cells. Now another thing, important thing that you will see that has been observed in natural population, that suppose an inverted gene sequence has undergone further inversion, an inverted gene sequence within the chromosome, that inverted gene sequence again it undergo inversion, so that a part of the inverted gene was involved in the second inversion as well. These type of inversion are called overlapping inversion. So if diagrammatically if you see, it is very easy to say this definition. A inverted segment undergo again second round of inversion that leads to an overlapping inversion. Let us see. So this is an overlapping inversion. You see two breaks has been occurred within this region, and that segment CDEF has been arranged invertly to form this inverted segment. Now this inverted segment again undergoes second round of inversion by causing break in this and this side, that leads to the formation of secondary inversion. You can see here a secondary inversion has been involved. This type of inversion is called as overlapping inversion. It is called overlapping inversion. So you can see that two breaks has been occurred to form a inverted segment, and that inverted segment is again involved in second round of inversion that leads to the formation of overlapping inversion. Now, cytologically, it has been detected that in case of humans, the most common type of inversion seen in humans is on your chromosome number 9. So, on chromosome number 9, in case of humans, the chromosome number 9 inversion occurs more in humans, especially if I say at which position of chromosome number 9 inversion has been observed. It is observed at inversion 9 p12 and q13. P refers to the short arm of chromosome number 9, q refers to the inverted segment of the long chromosome arm. So it has been observed that the most common inversion seen in humans is on chromosome number 9 at inverted position P12 and Q13, where P refers to the short arm of the chromosome while Q refers to the long arm of the chromosome. Okay, on that position, inversion has been observed in humans. Now, this inversion is generally to consider to have no harmful effects, but there is some suspicion that it could lead to an increased risk for miscarriage or infertility for some affected individuals. So, those who are having inversion at chromosome number 9, especially at its short arm at 12th position and long arm at 13th position, it is shown that there is a suspicion that it leads to an increased risk for infertility, having miscarriage for some affected individuals. Chromosomal inversion are also found as fixed differences between the species and as polymorphism within the species in many groups of animals and plants. So you find this type of inversions in the between the species also, and between the and polymorphisms within the same species and many groups of animals and plants. And what's the importance, why so much different types of inversions are found among the species without causing any harm effects? It leads to polymorphism. Actually, what does polymorphism means? Poly means many, morphism means morphological character. It simply means occurrence of two or more clear different forms, also referred to as alternative phenotypes in the population of a gene. So it means to say that with the help of chromosome inversions, we can make polymorphism within the same species, within the different species. And why what is the significance of inversion is that sorry, what is the significance of the inversion is that it will leads to rearrangement of genes that will help to that will actually help to put the genes into new associations for some desirable traits, for some desirable characteristics. So it has been written that the significance of inversion is that it results in rearrangement of genes. So by doing inversion, we are rearranging the genes in chromosomes, and therefore put that genes into new associations. And in such effect, in such situation, we can also find out the position effect also. Okay, so two things can be is observed while inversion occurs. One is polymorphism. That means uh, polymorphism simply means we are rearranging the gene segments, and by doing polymorphism in the non-essential part of the DNA, we can able to identify the differences between the same species. Okay, in this is the main reason that how DNA fingerprinting will help us to detect, okay, based on the polymorphism pattern between the same individual. Okay, secondly, inversion leads to inversion leads to rearrangement of the genes so that we can put the genes into new associations. Okay, and example of position effect. What is the position effect? Position effect is the effect on expression of the genes when its location in the chromosome is changed. So it simply means when we are doing inversion, we are making a different orientation of the arrangement of genes because they are arranged in the invert order. So we are changing the position also of that genes, and that's how position effect come into play. Position effect simply means effect on expression of a gene when its location in the chromosome has changed. So if the location of in the chromosome has changed, that is called as position effect. That means what will be the effect of the gene, what will be the expression of the gene if their position is changed. And an example of position effect is your female Drosophila heterozygous for X-linked recessive eye color mutant white. Now, this is a simple explanation of the position effect uh, that involves Drosophila heterozygous for X-linked recessive eye color mutant white. Now, the W plus W genotype, that means in one of this genome that there is a W plus allele, wild type allele, and another one is W allele, which is recessive allele. It results in a wild brick red eye color. Normally, Drosophila have a red eye color, and that is represented by W plus here, which is a dominant allele. Plus indicates the dominant allele. But however, if the region of the X chromosome uh, if the region of the X chromosome containing this wild type W plus allele is moved, or you can say translocated, so that it is close to a heterochromatic region, expression of that W plus allele is modified. Again, I am repeating, the W plus allele represents the brick red eye color feature, whereas the simple W allele is a recessive allele which will represents the white eye color feature. Now, if a organism, if the Drosophila have this W plus W genotype, that means in one of the chromosomes, one of the homologous pair of chromosomes, or you can say X chromosomes, because X chromosome comes in pairs in case of female, if it has W plus phenotype, if it has W plus gene in one X chromosome and W gene in another X chromosome, then due to the having of this dominant allele W plus, it will dominate the expression, dominate its expression, because dominant allele always express themselves whether they are found in the one chromosome or in both the chromosomes of the homologous pair of chromosomes. But if suppose that W plus allele position is changed, that means translocated, so that where it is changed, it is shifted to such a region which is close to the centromere, which is a heterochromatic region as a result of inversion. So, as a result of inversion, if within the X chromosome the W plus allele is shifted to close to a centromeric region, which is a fully heterochromatic region, inactivated region, fully coiled of histone proteins to make them coiled regions of chromosome, non-functional regions. So if the W plus allele is placed in that region, the expression of the W plus allele will be modified. Instead of having red color, the eyes will be variated or mottled with red and white patches, because the expression of the W plus allele has been hampered due to the changing in their position within the same chromosome as a result of paracentric inversion. So, following translocation due to inversion, the dominant effect of the W plus allele is reduced. So, this is called position effect. We are changing the position of a gene within the chromosome as a result of translocation or as a result of inversion, that leads to variation in their expression of that particular gene. So
and that's the example comes the W plus allele feature, which represents the brick red eye color of your Drosophila fruit fly. Now, Drosophila fruit fly, their X chromosomes have these W plus allele present. Well, it also contain W alleles. The W alleles represents the white eye color feature, which is the recessive allele. Now, if a female fly having the having the X chromosomes, one having a dominant W plus allele, and in another X chromosome of that homologous pair having W allele, well, due to the presence of the W plus allele, it is a dominant allele, it will express its feature, that is the brick red eye color feature. But if its position is changed as a result of inversion, as a result of paracentric inversion, if its position is changed and placed close to the chromosome, close to the centromere of the chromosome, when what happened, as the centromere is a heterochromatic region, fully coiled region that does not contain any essential genes. So if the W plus allele is placed close to the centromeric region, the expression of the W plus allele will be hampered, or it will be affected, and thus that type of fly will be having, even they have a W plus allele in their X chromosome, but still as they are placed within the close to the centromere, the expression of the W plus allele is modified. Instead of having red brick eye color, the eyes will be with variated or mottled with red and white patches. So, following translocation, following the movement of the chromosomes segment as a result of inversion, the dominant effect of the normal W plus allele is reduced. So, this was all about your inversion, and we have also seen that most of the case we have this paracentric inversion, where the inverted segment does not involve centromere, and this leads to a formation of this type of bridge formation, which eventually leads to the formation of such products which are actually half of them are sterile and half of them are functional. Okay, we have also seen overlapping inversions as well. In overlapping inversion, we have seen that how the inverted segment is involved again for the second round of inversion, leads to the formation of overlapping inversion. And cytologically, we have seen that the inverted segment will try to pair with its homologue by forming a loop in order to pair themselves, align themselves during crossing over, during synapsis. Okay, so that's all for this inversion. I hope you have understood this part. If you have any questions regarding this chapter, regarding this portion, you can comment me in WhatsApp group. I will try to explain with answer. Okay, the next video that I will be uploading a little bit later is your translocation. Okay.